Hi, my name is Rob McLeod from the University of Utah and the Scientific Computing and Imaging Institute. This is another in our series of documentation videos to show you how to use UncertainSCI in your simulation projects. This was one of a series we recorded at a recent modeling conference on functional imaging and modeling of the heart, but this is actually a project related to brain research in which we show the power of UncertainSCI to help predict sensitivity to parameters in the context of neuromodulation. We welcome our speaker for today, Sumita Ramprasad, to describe how she uses UncertainSCI in her research.
Thanks, Rob, and thanks for any cardiac people who are still here, showing up to a neuromodulation talk in a cardiac conference. You might wonder what I'm doing here. I know nothing about hearts, but with respect to bioelectricity and simulations of bioelectricity, there's a lot of similarities between cardiac models and the kind of models I'm going to show you today. And I'm going to look specifically at uncertainty of tissue conductivity and electrode locations. And basically, if you are working on anything that and have any interest in those parameters, whether it's in a brain or a heart or a spine, what I'm going to show you could basically almost identically apply. And also shows the versatility of the UncertainSCI package. All right, so I'm going to talk about two different types of brain stimulation. First one is TDCS, where we put electrodes on the scalp and we send weak currents through for moderate amounts of time. And that has effects on neural excitability. And there's a lot of clinical applications, such as motor rehabilitation after stroke and depression, and also non-clinical applications, such as improving working memory in healthy adults. And the second stimulation method that we'll look at is ECOG stimulation. And here electrodes are placed directly on the cortex, and short current pulses are given through pairs of electrodes. And this can be used for mapping of cortical regions in epilepsy patients before surgery, also as therapy, and in brain-computer interfaces. And main differences between these two techniques is ECOG is invasive, has a lot of risks compared to non-invasive TDCS, and it being invasive, it is of course much more accurate in targeting. And then in similarities, both have a lot of different positive effects on brain activity, but those effects are very variable and the mechanisms are not fully known. So simulation and optimization can improve our understanding of these techniques and hopefully improve the targeting that we use in experiments. So here's a little overview of how we do simulations for neuromodulation. And we start with describing physics. Here we just have a Laplace equation with two boundary conditions. And we take MRI scans of the head and segment those into different tissue types. And then we take the boundaries between those segmentations and we make a finite element model. And then to every element of the model, we assign a conductivity value, either a scalar value or a tensor using diffusion tensor imaging. Then we build electrodes on top of that model and we throw it into SciRun. And SciRun will solve the equation and present us with the solution that is the potential anywhere in the head model, potential on the nodes. From that, we can calculate electric field in all the elements and whatever else we might be interested in. And so we can do that for a given configuration of electrodes that we know, or we can try a bunch of different ones and try to optimize where we should put the electrode and send the currents to reach a certain target of the brain. And then we can apply that in the experiment and do some measurements. Now, in these simulations, there are several sources of uncertainty. And one is in the geometry of the models. So you have the very wavy structure of the cortex, right? It's kind of like a cauliflower. And there's a lot of very narrow areas in there. Similarly for the CSF, there are very thin areas that are hard to capture accurately with an MRI scan and also difficult to model with finite element models. And then there are the tissue conductivities, and we have values from literature that come from measurements that people have done with animals or with human cadavers. And as you can imagine, it's quite difficult to do that in vivo. In humans, they tend to not like that. But there are actually a lot of factors that influence conductivity, such as temperature, frequency of stimulation, varies between individuals and within individuals. And then another thing that we have uncertainty in is the location of the electrodes. So what you see here on this image is a post-op CT of electrocorticography grid implanted in the brain, registered to a pre-op MRI. And you can see that the electrodes, which are shown here in as red little circles, end up inside the brain. And clearly that's not where they are, but due to surgery, the brain actually changes a little bit in shape. And that causes this shift that you see here, which means that we have to move the electrode somewhere, and there's a lot of uncertainty in that. Now, what we're going to talk about specifically today is how to quantify uncertainty in tissue conductivities in TDCS and ECOG simulations. So first, we have this model. We put 2 electrodes on roughly the motor cortex bilaterally, and we're going to simulate TDCS with 1 milliamp of current in ski run, and we will calculate the electric field strength. This model has 6 different tissue types. So we have white matter, gray matter, cerebrospinal fluid, skull, and scalp. There are also eyes, but you cannot see them here. And then, of course, we have the electrodes. Now, we are going to model these conductivities not as single values, but as distributions. And we are using the range of these values comes from a study that was published. And we're choosing the beta distribution here with alpha and beta equal to 3, because we want a roughly normal distribution with only positive values. Clearly, if you have better information about what that distribution should look like, that could be something else. So now let's look at how we model this in UncertainSCI. So here's the code to set up this problem in UncertainSCI. And on the top, we define these ranges of conductivity values that are in the table. So we just give them min and max. Then the second part describes the distributions. So we're setting alpha and beta. And then for each parameter, we're modeling a beta distribution, which UncertainSCI just gives us. That's just a function that's available. And here we're setting alpha and beta equal for all the 5 conductivities, but of course you can vary them if you have that information. And then finally, we use the tensorial distribution function to combine all these distributions into one. And then the next part is to initialize a PCE object. So we need to know the dimension of the problem. And in our case, the dimension is 5. We're varying 5 different parameters in our simulation. And then we need to set the polynomial order. And we're choosing here to be 8. And then we can build this PCE object. Now, the next step, now that we have set up the distributions of the samples that we want to model, we can actually generate the samples. And really the only part of what I'm showing you that you really need is this yellow part, PCE.generateSamples. That function will produce PCE.samples, which is an array that has, for every iteration, 1 row consisting of a sample for every parameter that we're trying to change. So in this case, it's number of iterations by 5, and the number of iterations is determined by the order and the dimension of the problem. Now, once we have the samples, here I'm actually saving them out to a text file in case I want to know what they are. And then the rest of this section is a little bit of a fail-safe. If you have a fairly expensive model, which in my case I have, so this is a 4.2 million elements head model. And if you're running a lot of iterations, so either have a high dimension or high order, then there's a chance that your software might crash at some point, and then basically UncertainSCI would have to start over, which obviously is something that would not be great. So a solution to that is to just build in some some fail safes basically. And here we just check if there's already a parameter file here, we'll load it. And if not, we'll generate new samples. Now, once we have the samples, the next thing we do is to feed those to SCI and run the simulations. And what UncertainSCI needs to do its calculations is all the solutions for all the iterations, for all the samples that we're taking. So we need to prepare a matrix that has a size of number of iterations by number of elements in the model, or number of values in your solution, whatever it may be. So we prepare that array here, and then we loop over all the samples. So we loop over pc.samples, and we take for every loop one row of this array. And so each row is 5 conductivity values. And we feed those into this run SCI function. So let's look at the run SCI function. So this is the part of the code where we're going to actually do the simulations. And we're going to start with setting up some paths. First, we have the SCI executable and the flags that we want to run. And in this case, I'm using the S flag, which tells SCI that I'm going to give it a Python file. So I'm giving it a file with some Python instructions, and then it will run that. Then here I set the SCI network that I want to run. So if you have a different problem that you're trying to solve, this is the line that you would change. You change it with your own network. And then we're setting the output file. And again, this is not necessary, but it's more of a fail-safe, because let's say your software crashes somewhere after 200 iterations, UncertainSCI would have to start over because it needs all the solutions in memory at one time, which also, by the way, means you should be a little careful on what machine you run this, because it might cost a lot of memory. But in case it crashes, if you save out all the solutions as separate files, you can just reload them, which makes life a lot easier. So this part, this if statement here, also checks if the file already exists, reload it. If not, let's go ahead and actually run SCI. So let's say we're going to run SCI. Then where we end up is here. So we take this parameters array, which is that one line of the matrix that we sent in. And we take all the 5 separate values out, and we make strings out of them so that we can write them into a text file. This next section here is where we write the text file with Python code to run SCI. So let's first check what our SCI network looks like. And this is a basic TDCS solving network. It loads the head model. It sets the conductivities for all the different tissue types. It loads the electrode potentials, and then it solves the Laplace equation, and it produces potentials and electric fields. Now, what I've done here is split up all the conductivities into separate create matrix modules. And each of those modules looks like this. There's just a single value. And here there's the default value for white matter is 0.14 Siemens per meter. And we can set these default values for all these different conductivities. And then here we append them into one big matrix, and we can convert the tissue indices to conductivities and then continue with the simulation. Now, if I would run it like this, it would be a standard TDCS simulation with single conductivities for every tissue type. So now how do we actually get this to change for all these different samples? And we can do that by preparing a Python file, and that will look like this. And we use some SCI specific Python functions. And here we tell SCI to load the network, and we give it the path of the network. And then we tell it set module state of module create matrix 6 to this value here. Now, create matrix 6 is this one that I have open here. So that's the white matter. And you'll see these values here change with these 5 lines that I have, and they don't really mean anything. Basically, SCI just keeps numbering whatever you put in there. So you check what the number is of the matrix that you care about, and you can just change the value here. And then here we're also changing the output file. And then we tell SCI to execute all modules, then it will close. So basically what we need to do in UncertainSCI is to create this file that will load the network, set all the connectivities, and run. Now here we have the text that does that. So here we write a text file, and we write all those lines that we just saw, and that produces this file here. And then once we have that, we can just call SCI-run, have it load the file and execute. And then we load the solution and we spit it out. So then we've run all these, we've created our samples, we've run all the simulations, we gather them into one array and then put them into the PC object. And now we can do the post-processing. And you've seen this before, Jess has talked about this. So we can look at mean, standard deviation, total and global sensitivity, quantiles, and we can save it out to MATLAB or whatever you prefer. So now let's look at the results of the calculation that we did. So here you see the brain surface and the electrodes kind of hovering above because they're on the skin, but we are obviously mostly interested in the brain. So here we see the mean electric field strength on the brain, and as you can see, the values are high under the electrodes, and that's exactly what we would expect. So this is basically identical to a standard CDCS simulation without any UQ. You take the average values and you run it, this is what you get. Then when we look at the standard deviation, we see that looks a little different, but it is high where electric fields are high, which also is what we would expect. Now we can also look at what happens inside of the brain. And we're here taking a coronal slice through the center of the electrodes. And we're seeing that basically the mean and standard deviation have very similar distributions, and they look like we would expect them to look. Now, what's really interesting, right, is the sensitivities. And as was mentioned before, we have one sensitivity value, one total sensitivity value for each parameter that we changed. And here you can see that, for example, for the CSF, nothing happens really. It seems very uninteresting. And that makes sense because we actually only varied the CSF by a very small amount. We have more knowledge about the range of values for that tissue type. And we see that for the skull and scalp, the values are highest close to electrodes. For the gray matter, it's basically everywhere. And then when we look at the white matter, we see the highest values are very far away from the electrodes. The lowest values are close to the electrodes. And that seems a little odd, but what you have to remember is that these sensitivities tell you the amount of the standard deviation that can be attributed to this parameter. So if we look at the standard deviation, right, it's very small in that same area. So even though this sensitivity looks big, large, the actual effect that this has in this area is very small. So it can be difficult to interpret these values, and you really need to think about it well. Now also we will look at the cut through the model again, same for the CSF, right? Really not interesting, but now for the other tissue types, we see a lot more happening. And so white matter primarily affects white matter, gray matter affects gray matter, skull affects skull, and scalp affects everything. Now let's switch to the second problem, which was the ECOG stimulation. In this case, we have still gray matter, white matter, and CSF, but we've taken off the scalp and the skull because the electrodes are directly placed on top of the CSF. Again, we use the same values, same distribution to model this, but now we're also going to vary the electrode locations. So the electrodes are modeled as point sources, and we're going to choose cathode and anodes from this group of available locations. Then for the UncertainSCI code, this is very similar to what we looked at before. Right, so we have the same conductivity range set up here, the same beta distribution set up here. The only difference is now that for the locations, we use a discrete uniform distribution and give it the number of samples to pick from. And everything else is basically identical. So now we look at the results, and in this case, we are looking at potentials instead of field strength. And we can see here that for the mean, we see potentials that are high close to the electrodes, which is exactly what we would expect. And also the standard deviation is high there. But in this case, a little bit further from the electrodes, there's really nothing happening. And if we look at the total sensitivities, the same happens. So really, the only area where something interesting happens is here in this plane of the electrodes. But what we can clearly see that in that area of interest where the standard deviation was high, only the cathode and anode have very large contributions to the uncertainty in the resulting potentials. Now, again, we can look at a slice through the model, through the centers of the electrodes. And in this case, we see very similar results as for the surface, where with TDCS, we learned a lot from cutting through the model. It was a very different result than seeing the surface. In this case, it doesn't really give us that much more information. So that was total sensitivities. We also have global sensitivities. And the interesting thing about that is that we can look at interactions. And here I'm showing all the interactions for the ECOG model. So as you can see here, we have, for example, white matter with CSF, or white matter with cathode, white matter with anode, et cetera, from primary, secondary, tertiary, all the way up to 5 different parameters combined. And for the TDCS case, we can do the same thing, but if you look at the part that I marked in red here, that is the part that belongs to the primary values, so not the interactions. And for the ECOG case, the interaction part is very big and the primary values are very small. For TDCS, it's completely the opposite. So this shows that while the simulations are fairly similar, when we start looking at electrode locations, results are very different. The last thing I want to talk about is comparison to other software that can do similar quantifications. And one software package that is suited for what we're trying to do here is UncertainSCI. And like UncertainSCI, this is a free Python-based software package, and it uses polynomial chaos. It has 2 options, either a collocation or a spectral method to do the sampling. It can also connect to other software, to SciRun, similarly as I showed for UncertainSCI. However, it is limited in the size of the model that you can put into it. So the HAT model that I used before had 4.2 million elements, and UncertainSCI was totally happy with that. But when we threw that into UncertainSCI, it crashed. And this is basically because UncertainSCI was developed for neural models, which are much smaller, and it doesn't handle these very big models very well. So to still be able to compare them, what we did was take a slice out of the model and only put that into UncertainSCI and calculate the uncertainties on that slice. And what we saw was that the results for UncertainSCI and UncertainSCI were almost identical. So that's a good validation of what UncertainSCI is doing. Then second thing we did was to look at efficiency. And what we're doing here is varying the order, the polynomial order that we use to run the UncertainSCI or UncertainSCI. And we varied it here from 1 to 19. And looking at this first plot, we're looking at the number of SciRun runs. So you can see that UncertainSCI needs more iterations for the same order than UncertainSCI does. And then you would expect that the runtime would scale in a similar way, but that's actually not the case. So while UncertainSCI scales linearly with order, UncertainSCI goes up much faster and actually becomes quite unfeasible to run for higher orders. Similarly for RAM usage, so up to about order of 10, they're fairly equal, but then UncertainSCI goes off the charts, and yeah, you might not have 80 gigs available. For CPU usage, UncertainSCI slowly increases with order, and UncertainSCI just uses whatever you have available. So in conclusion of all of this, we saw that UncertainSCI accurately and efficiently quantified uncertainties in simulations of brain stimulation. And with respect to these specific cases, we saw that TDCS simulations are mainly affected by scalp and gray matter connectivity, while ECOG stimulation was most strongly affected by the anode and cathode location and had many interactions with all the different conductivities. And we will do in future work, we will continue looking at these stimulation modalities and then also investigate effects of uncertainty in the geometry of the model and also in white matter anisotropy and electrode location for TDCS, which I didn't show here, and for other stimulation modalities. Oh, I forgot to mention this, very bad. The ECOG simulations were done by Chantal Charlebois, who is also here. So if you have any ECOG questions, she is available.
Yeah. We hope you've enjoyed this video and been motivated to try and incorporate uncertainty quantification into your solutions, into your software simulations, no matter what type of application they might have. And we hope you'll be tempted to try our UncertainSCI toolset, which can help you incorporate this sort of analysis into virtually any type of simulation. Please check our website, check the additional documentation, and reach out to us and let us know how we can help you enjoy the benefits of UncertainSCI in your simulation research.